In dark reaction, there will be fixation of carbon dioxide, making use of ATP and NADPH2. The method of carbon dioxide fixation in plant is different. Method of carbon dioxide fixation in plants is different. One method of fixation of carbon dioxide, that is C3 cycle. It is also called as C3 pathway. It is also known as Calvin cycle. This is one method. This is one method. Then another method, most of the plants where the carbon dioxide fixation occur by this method only, C3 pathway only. Okay, 97% of the plant species in which this carbon dioxide fixation occur, by which the synthesis of carbohydrates or sugar occur, mainly by C3 pathway. In some of the plants which are xerophytes, which grow in dry conditions, desert-like conditions, okay, where water availability is comparatively less and high temperature will be there, especially in tropical and subtropical regions of the world, there another method of carbon dioxide fixation occurs, that we call it as C4 cycle, or it is called as C4 pathway, or it is known as hat and slat pathway. Hat and slat pathway. These are the two main pathways by which there will be carbon dioxide fixation to synthesize the sugars or carbohydrates. Okay, there are some plants, okay, they are xerophyte, true xerophytes. True xerophytes they are. Okay. These xerophytes like Opuntia, cactus, Opuntia, and pineapple, galante, bryophyllum, etc. In these two xerophytes, you can see another pathway of carbon dioxide fixation, that is called a CAM pathway. CAM pathway. Okay, so this CAM pathway refers to crassulacean acid metabolism pathway. Crassulacean acid metabolism pathway. This pathway is not there for our syllabus, so we have to learn mainly C3 and C4 cycles. That we have to keep it in mind. Crassulacean acid metabolism pathway, seen mainly in xerophytes, okay, like Apothecia, cactus, pineapple, bryophyllum, calonche, etc. So we will begin with first one, that is C3 cycle, or what you have, Calvin cycle. This is one of the important method of carbon dioxide fixation in plants. See, this pathway was worked out by an American plant physiologist by name Melvin Kelvin. How carbon dioxide is fixed into a sugar carbohydrate. How the carboxylation can result into the synthesis of sugar. This was worked out by an American plant physiologist or biochemist, okay, by name Melvin Kelvin. He was working at the University of California. Along with him, his co-workers, his friends, such as Andrew Benson and James Basham. Andrew Benson and James Basham, they assisted in finding out the pathway of carbon dioxide fixation. They did research on unicellular green alga, that is chlorella. unicellular green alga, that is chlorella. They use even scanerismus. They use even another green alga, that is scanerismus. Okay. What they did, see, carbon dioxide is used during dark reaction, you know that. Okay. These dark reactions are also called as Blackman's reaction, you know that. Blackman's reactions. F.F. Blackman, who first time found out that there are some set of reactions which do not make use of light. They can occur during night time, but also can occur during daytime, but without using sunlight. Dark reaction, that doesn't mean that they should occur only during night time. They can occur during daytime also, but they don't make use of radiant energy. Okay, so carbon dioxide is used by the plant to synthesize the carbohydrate. So they used radioactive level carbon. Normal, you know that is C12, atomic number 6, atomic weight 12, C12, normal carbon dioxide. But carbon dioxide, which they use mainly to trace the pathway, to find out the pathway of carboxylation or carbon dioxide fixation, to synthesize the sugar, they use C14 level carbon dioxide to find out the sugar is synthesized from which what we call source, and how are the steps involved in the synthesis of the sugars, right? Suppose if you use this carbon dioxide which is level with C14, when the sugar is synthesized from this carbon dioxide, this sugar will also have C14. So tracing the pathway. So here they used what we call the techniques such as chromatography. They used the technique that is called chromatography, and they used another technique that is called autoradiography. By using the technique, okay, they found out the complete reactions mechanism of sugar synthesis by using the carbon dioxide. Okay, they were working at the Lawrence Radiation Laboratory, University of California. They worked on chlorella mainly, and also they used scanerismus. Using radioactive level carbon, okay, they found out this complete mechanism of reactions, set of reactions. You know that these reactions, carbon dioxide fixation steps, these occur in the stroma of chloroplast. Stroma of chloroplast. These chemical reactions, which are there here, these chemical reactions, different set of chemical reactions are there. These chemical reactions are catalyzed by enzymes, mediated by enzymes. These enzymes are present in the stroma or what we call matrix of chloroplast. That's why dark reaction occur in the stroma or matrix of chloroplast. Why dark reaction occur in the stroma or matrix? Why not in grana? In grana, dark reaction enzymes are not present. In stroma or matrix, dark reaction enzymes are present. That's why these reactions occur in stroma or matrix of the chloroplast. So try to remember this. So that is why this C3 pathway or it is having other name, Calvin Benson cycle. One more name you can give, Calvin Benson and Persham cycle. As his friends, his co-workers also assisted in finding out the complete set of chemical reactions resulting in the synthesis of carbohydrate. That's why this pathway is known by different name. Okay, it is also referred as reductive pentose pathway. reductive pentose pathway RPP. Now listen, why it is called C3 pathway? The name C3 pathway is given. In this reaction or in this cycle, when carbon dioxide is fixed, the first stable intermediate carbon compound, remember, the first stable intermediate compound formed during this cycle of this pathway, that is 3 carbon compound. The 3 carbon compound is 3PGA. Remember this, very, very important, boys and girls. This is 3-phosphoglyceric acid. 3-phosphoglyceric acid. 3-phosphoglyceric acid is the first stable intermediate compound formed in this pathway. That is why it is called as C3 pathway or C3 cycle. The plants which exhibit C3 cycle or C3 pathway, they are called as C3 plants. They are called as C3 plants. So majority of the plants, they exhibit C3 pathway. Few plants can exhibit C4 pathway. Very few xerophytic plants adapted to desert conditions can exhibit CAM pathway. So these are all different methods of carbon dioxide fixation resulting in the synthesis of carbohydrate during dark reaction. That is the second set of reactions which occur in the chloroplast after the light reaction, making use of assimilatory powers of light reactions such as ATP and NADPH. Right? So now, the main mechanism. What are the steps involved in C3 pathway? Steps involved in C3 pathway. Kelvin cycle C3 pathway enzymes are present in stroma or matrix of chloroplast. That's why this cycle occurs in stroma or matrix of chloroplast. Remember. So what are the different steps of this C3 pathway? The steps are carboxylation. First step is carboxylation. It is also referred as carbon dioxide fixation. Then the second step is reduction. Second step is reduction. And the third step is regeneration of RUBP. Regeneration of RUBP. RUBP here, ribulose 1,5-biphosphate. Ribulose 1,5-biphosphate. This Kelvin cycle is very, very important for 5 mark for theory exam. And on this, many neat question you can expect. That very important for theory exam. Photosynthesis chapter 1 5-mark guarantee. Usually respiration 1 5-mark guarantee in theory exam. So these two chapters if you perfect, 10 marks will be your point. So these are the 3 steps. One by one, these steps we will learn. Then afterwards, we will draw that cycle, schematic pathway. So the different set of chemical reactions of these 3 steps occur in cyclic manner. It occurs in cyclic manner. That's why it is called cycle. Remember the alternate names of this pathway. So first step is carboxylation. The step where carbon dioxide is used. Now listen, these reactions occur in the stroma of the chloroplast. The starting precursor, what you have, that is RUMP. It is a 5-carbon compound. RUBP is produced from RUMP. Okay, it is a 5-carbon ketone sugar. It is a 5-carbon ketone sugar. This is primary acceptor of carbon dioxide. This is only will accept the carbon dioxide. This 5-carbon ketone sugar concentration is comparatively lower. That is why this will be produced from this RUMP. So RUMP is ribulose 5 monophosphate. Phosphate group is attached to the 5th carbon. It is a 5-carbon compound at all. 5-carbon compound. This phosphate group is attached to the 5th carbon. That is the number ribulose 5 monophosphate. Okay. This is converted into ribulose 1,5 biphosphate. So conversion of RUMP to RUBP require energy. That energy is provided by the ATP, which is a product of light reaction. One molecule of RUMP on phosphorylation, okay, where ATP is used here, it gives rise to RUBP. It gives rise to RUBP. Okay, ribulose monophosphate, one phosphate is added. Here, how many phosphate will be there in ATP? 3 phosphate. Adenosine triphosphate. See, adenosine triphosphate, it will be like this. Okay, this will be separated. This can be added to it. Monophosphate will become biphosphate. 2 phosphates will be attached. Where they are attached? First carbon and 5th carbon. Ribulose 1,5 biphosphate. Okay, ribulose 1,5 biphosphate. By 2, it is also called diphosphate. In some books, there will be bisphosphate also. That is also correct. So this is the 5-carbon ketone sugar. This ribulose 1,5 biphosphate is primary carbon dioxide acceptor. Means the compound which accepts the carbon dioxide is ribulose 1,5 biphosphate. Remember this. It is also referred as RUDP also. RUDP means diphosphate. But same meaning, 2 phosphates will be there, right? So here, what you will get here? If this phosphate is added here, how many phosphate will remain now? 2. So product can be ADP, adenosine diphosphate. Okay. In Kelvin cycle or in dark reaction, to synthesize one glucose molecule, there is need of 6 carbon dioxide molecules. Is it or not? In reaction also we have written photosynthesis reaction. 6 carbon dioxide molecules will be used. 12 water molecules undergo splitting, resulting in the formation of 1 glucose molecule. 6 molecules of oxygen liberated and 6 water molecules are formed. Right? So here, 1 molecule of carbon dioxide is fixed by 1 molecule of RUBP. 1 molecule of RUBP fixed, 1 molecule of carbon dioxide. That means to fix 6 molecules of carbon dioxide, how many molecules of RUBP we need? 6 RUBP we need. 6 RUBP or RUDP we need. That means here we can have 6 molecules of ribulose monophosphate. Okay. On addition of 6 molecule of ATP, that is phosphorylation. Okay, phosphate group is added to it, and there will be a synthesis of 6 molecule of high carbon primary carbon dioxide acceptor, ribulose 1,5 biphosphate. So this now acts as the primary carbon dioxide acceptor. It fixes the carbon dioxide. That step is called as carboxylation. Okay, so we'll write that. See. one time one RUBP fix one carbon dioxide molecule. At that time it will not fix it. One time one RUBP fix one carbon dioxide molecule. That means how many times this reaction should be repeated? 6 times. Remember, to complete this C3 cycle or Kelvin cycle, if this is the cycle, we have to take 6 turns to complete one Kelvin cycle. Remember, to complete one Kelvin cycle, it has to take 6 revolutions, 6 turns. In one turn, one carbon dioxide is fixed by one molecule of RNA. So 6 turns, 6 molecule of RBP fix 6 molecule of carbon dioxide to synthesize one glucose molecule. Remember, boys and girls. So every time I did not mention 6, one I will mention to avoid confusion. Remember how many turns it requires? 6 turns for the synthesis of one molecule of carbohydrate at the end of Kelvin cycle. The main product of this dot reaction is sugar, glucose. Right? So now this RUBP ribulose 1,5 biphosphate. This you don't remember. Okay. It is going to fix carbon dioxide. This step where ribulose 1,5 biphosphate fix the carbon dioxide by the catalytic activity of the enzyme. This step itself is called as carboxylation. Okay. So here, fixation of carbon dioxide. This fixation is catalyzed by Rubisco enzyme. Very important enzyme. Rubisco enzyme. Rubisco, ribulose, biphosphate, ribulose biphosphate, carboxylase, oxygenase enzyme. This Rubisco has dual function. Dual function. Dual function means it can act as RUBP carboxylase. It can act as a ribulose biphosphate carboxylase enzyme when relatively carbon dioxide concentration is more. When relatively carbon dioxide concentration is more. When relatively carbon dioxide concentration is more, relative concentration of carbon dioxide when it is more, okay, then this fix carbon dioxide. This enzyme help in fixing the carbon dioxide. So that's why it is carboxylase enzyme. The function of ribulose biphosphate carboxylase oxygenase enzyme dual nature. When the relative concentration of carbon dioxide surrounding the leaves is more, then it acts as RUBP carboxylase fixed carbon dioxide. In the surrounding of the leaves, if carbon dioxide concentration is low, relatively low, it is relatively low, okay, and oxygen concentration is relatively high, oxygen concentration relatively high, then this RUBP carboxylase now it can act as RUBP oxygenase. Instead of fixing carbon dioxide, it starts fixing oxygen. Remember, that is why it has dual nature. If CO2 level relative concentration is more, no problem at all. Rubisco can catalyze carbon dioxide fixation. Suppose relative concentration of carbon dioxide is low, oxygen is more, then instead of fixing carbon dioxide, it starts fixing oxygen. So it is named as regular biphosphate oxygenase. Okay, so when it fixes the oxygen, the plant performs photorespiration. The plant performs photorespiration that we will learn little later on. C3 plants can perform photorespiration. C3 plants can perform photorespiration, whereas C4 plants avoid photorespiration. C4 plants do not perform photorespiration. Because of photorespiration in C3 plant, photosynthetic yield can be reduced. Remember, photosynthetic yield can be reduced because of photorespiration. We will learn little later on. Okay, so now regular biphosphate, 1,5-biphosphate, fixed carbon dioxide. Okay, so we know that 6 molecules of regular biphosphate fix 6 molecules of carbon dioxide. That you should remember. Okay, so now in one turn, how many carbon dioxide is fixed? One. Okay, so I did not write every time this is 6. Now, this is 5 carbon compound, right? And this is how many carbon compound? 1 carbon compound. Fixed fuse. Okay, you are going to get 6 carbon compound here. This is unstable compound. Unstable 6 carbon compound it is. You will get the 6 carbon unstable compound. Okay, 6 carbon unstable compound. This 6 carbon unstable compound name is 2-carboxy 3-keto arabinitol 1,5-biphosphate. 2-carboxy 3-keto arabinitol 1,5-biphosphate. That is the 6 carbon unstable compound form. This 6 carbon unstable compound will split. As it is unstable compound, it will split. Remember, this Rubisco is abundant protein in the chloroplast. It is the most abundant protein in the chloroplast which catalyzes carboxylation in C3 plant. Okay, 16% of the chloroplast protein is formed mainly from this Rubisco. So this 6 carbon unstable compound, okay, 6 carbon unstable compound splits. It splits into exactly half the carbon compound. Exactly half the carbon compound means 3 carbon compound will get. See here, 6 molecule of 6 carbon compound, that remember. 6 molecules when you will get, when 6 molecule of RUBP fix 6 molecule of carbon dioxide, then we will get 6 molecules of 6 carbon unstable compound. Keep it in mind. Okay. So now 6 carbon, 6 molecules of unstable compound, that is 2-carboxy 3-keto arabinitol 1,5-biphosphate split by making use of water, and you are going to get a 3 carbon compound. That is the first stable intermediate compound formed in this pathway. It is 3-PGA, 3-phosphoglyceric acid. 3-phosphoglyceric glyceric acid. It is also written as 3-phosphoglycerate, both 1 and 7, 3-phosphoglycerate or 3-phosphoglyceric acid. Okay, 3-carbon compound, right? This is 6-carbon unstable compound. How many molecules? 6 molecules. Now, how many molecules of 3-carbon compound we will get here? 12 molecules of 3-carbon compound we are going to get, because this is here 6 molecules of 6-carbon compound. 6-carbon compound splits into 3-carbon compound. Number will double or will get reduced. 3-carbon compound, how many molecules will be there? 6 carbon, 6 molecules. 3 carbon, how many molecules? 12 molecules. So we will get 12 molecules of 3-phosphoglyceric acid, that phospho group attached to third carbon. That's why it is 3-phosphoglyceric acid. This is the first stable product formed. This is the first stable product formed. That is why this pathway is called as C3 pathway or C3 cycle. Remember, why Kelvin cycle is also called as C3 pathway? Because the first stable compound formed after carboxylation step is 3-phosphoglyceric acid, 3-carbon compound, hence it is called as C3 pathway. So we got 12 molecules of 3-phosphoglyceric acid. Okay, so these are the sequence of reactions that are taking place in the stroma or matrix of the chloroplast. Okay, so these are all different reactions which comes under carboxylation step. Carboxylation step. Clear these steps now? If you remember the name, okay, otherwise you leave it. That you remember 6 molecules of 6-carbon unstable compound. That much you remember. Remember, Rubisco is one of the most important enzymes which catalyze carboxylation in C3 plant. With abundant protein present on the earth, 16% of the chloroplast protein is constituted by mainly Rubisco. So this is all about the first step, that is carboxylation, that has been resulted into the first stable intermediate compound, that is 3-carbon 3-phosphoglyceric acid, 12 molecules we got. Okay, so in the first step, that is phosphorylation of ribulose monophosphate into ribulose biphosphate, there how many ATPs have been used? Total 6 ATPs have been used. 6 ATPs have been used, that remember. Okay. Now the second step, that is reduction. In the reduction process, what will be continued here? This 12 molecules of 3-PGA, it is a 3-carbon compound. It is a 3-carbon compound, right? So 12 molecules of 3-phosphoglyceric acid now will undergo phosphorylation. Will undergo phosphorylation. Phosphorylation. This is second time ATPs are being used here. First time in the first phosphorylation RUMP to RUBP, 6 ATPs have been used. Okay, so here 12 molecules of 3-carbon compound, that is 3-phosphoglyceric acid on phosphorylation. It is going to produce 12 molecules of 3-carbon compound, 1,3-dipga. It is 1,3-diphosphoglyceric acid. 1,3-diphosphoglyceric acid plus 12 ATP. So what we want? 12 molecules of 1,3-diphosphoglyceric acid, biphosphoglyceric acid, whatever you write, right? Now, this molecule, this organic compound, okay, is a 3-carbon compound, 12 molecules of 1,3-diphosphoglyceric acid, a 3-carbon compound will be reduced now. Actual reduction step. This is actual reduction step. Here, phosphorylation occurred that has resulted in 12 molecules of 1,3-diphosphoglyceric acid. Now, actual reduction. So, what happens is 12 molecules of 1,3-dipga will be reduced by the strong reducing power which is produced from light reaction. So, which is that strong reducing power? NADPH. Okay. So, at the end of light reactions, ATP and NADPH2 are produced. Okay. So here, in the first phosphorylation step, 6 ATPs have been used, and in the second phosphorylation step, 12 ATPs have been used. So total ATPs used here, 80 ATP. Remember. Okay. Now, NADPH2. 12 NADPH2 are produced at the end of light reaction. At the end of light reaction, remember, for every 2 water molecules split, 1 oxygen is liberated. How many water molecules are split as per the reaction? 12 water molecules. That means how many oxygen it can liberate? 6 oxygen it can liberate. For 1 water molecule, 1 NADPH2 is produced in light reaction. How many water molecules are used? 12 water molecules are used. Okay, on splitting, it can lead to how many NADPH2 synthesis? 12 NADPH2 synthesis. That you remember. Okay, so the number of the ATP synthesized at the end of light reaction that has been explained by chemiosmotic hypothesis that has been taught in online class. Okay, so chemiosmotic hypothesis result in the synthesis of ATP molecule. That remember. Chemiosmotic hypothesis only result in the synthesis of ATP molecule, right? So here, 12 molecules on NADPH2. This is reduction. Enzyme names I'm not giving. One enzyme, that is Rubisco, you want to remember. Each step is catalyzed by the enzyme. That is not compulsory, that is not required. That's why I'm not giving. Because remembering all the enzymes, it creates confusion. Mistakes you can do. You will avoid that. Carboxylation step enzyme you want to remember. That's one. Okay. So 1,3-diphosphoglyceric acid, 3-carbon compound will be reduced by reducing power, that is reduced nicotinamide adenine dinucleotide phosphate. Okay. So now we are going to get 12 molecules of 3-carbon compound, that is 3-PGAL. 3-PGAL. This is referred as 3-phosphoglyceraldehyde. 3-phosphoglyceraldehyde, 3-carbon compound, 12 molecules will get. That remember. 12 molecules will get. Okay. Time being, you remember, reduction step is called as glycolytic reversal. It is called as glycolytic reversal. The steps of these, what I am discussing, these steps occur even during glycolysis. In glycolysis, it is a step of respiration. That is next chapter, respiration in plant, where we will learn. In glycolysis, glucose 2, 3-PGAL is formed. Starting compound glucose, and here PGL is formed. Here, in this reaction, from PGAL, glucose is formed. Reverse of glycolysis. Glycolysis is a step of respiration that we are going to learn in the next chapter. During glycolysis, a 6-carbon glucose is split. The intermediate compound, what you are going to get, is 3-phosphoglyceraldehyde. But here, reduction step in C3 cycle. Reduction step in C3 cycle, what happens? From 3-PGAL, what is synthesized? Glucose is synthesized. So it is a reverse of glycolysis. In glycolysis, glucose to 3-PGAL. In this reduction step, 3-PGAL to glucose. That's why it is called glycolytic reversal. Remember this, glycolytic reversal. Okay, so we got 12 molecules of 3-phosphoglyceraldehyde. Right? Now, these molecules will take part in sugar formation. Sugar formation. Okay, out of the 12 molecules, 2 molecules of 3-PGAL are involved in sugar formation. See, 2 molecules by means PGAL, how many carbon compound? 3 carbon compound. 2 molecules, 3 carbon, fused means what is formed? 6 carbon. That is sugar. Then how many left here? 2 are used in sugar formation. How many left out? 10. 10 molecules of 3-PGAL are involved in last step. What is that last step? Regeneration of RUBP. RUBP is involved in carboxylation. I told in one turn, one RUBP fixes one carbon dioxide. So to continue that cycle, what we need? RUBP we need. Without RUBP, no carboxylation. That's why regeneration of RUBP is essential. Okay, so remaining 10 molecules of 3-phosphoglyceraldehyde are involved in regeneration of RUBP, ribulose 1,5 biphosphate. Remember, the sugar formation steps occur in the cytoplasm of a mesophyll cell. Cytoplasm of a mesophyll cell, sugar formation. Regeneration step, as usual, where it occurs? Stroma. Carboxylation, reduction, regeneration usually occur in stroma or matrix of chlorophyll. Whereas sugar formation, where it occurs? In the cytoplasm of mesophyll cell. Remember. So in this C3 cycle or in this dark reaction, how many ATPs have been used here? 18 ATP. How many NADPs have been used? 12 NADPs. So remember this. Now listen. We will learn sugar formation. Afterwards, we will go to regeneration. So how sugar is formed? Okay, sugar formation. Now listen. How many molecules of 3-PGAL are involved in sugar formation? 2 molecules. Okay, 2 molecules of 3-phosphoglyceraldehyde. Okay, this is an example of 3-PGAL. Is an example of triose, 3-carbon triose phosphate. Remember, it is an example of triose phosphate. 3-carbon, triose means 3 carbon, pentose means 5 carbon. Phosphate, phospho group is there, so that's why it is triose phosphate. Now, out of the 2 molecules, 1 molecule of 3-PGAL isomerize isomerize. Its isomer is formed. 3-phosphoglyceraldehyde. 3-phosphoglyceraldehyde is 3-carbon compound. It isomerizes into its isomer. Isomer is also a 3-carbon compound. It is DHAP. Remember this. This is also a triose phosphate. DHAP is also a triose phosphate. So here, this DHAP, what is this? Dihydroxyacetone 3-phosphate. Remember this. Dihydroxyacetone 3-phosphate. Dihydroxyacetone 3-phosphate. This is a 3-carbon compound. So one molecule of PGAL as it is, one molecule of PGAL has been converted into its isomer. What is that isomer? DHA. Remember. Okay. So two molecules of PGAL which were formed in the stroma of the chloroplast, these are transported out of the chloroplast into cytoplasm of the mesophyll cell. In the cytoplasm of the mesophyll cell, what is happening? Sugar formation. Okay, so now you see here now. One molecule of PGL is there. One molecule of 3-PGAL, 3-carbon compound. And one new molecule isomer is formed. What is that? DHAP, dihydroxyacetone 3-phosphate. Undergo condensation and fuse. Undergo condensation. Okay. Aldolase enzyme is involved. I'm not mentioning many of the enzymes. Okay. Aldolase enzyme is involved. Aldolase enzyme. And now, what we will get? Okay. Aldolase, that is previous step. You leave it. Aldolase, you leave it. Okay. Now, product is fructose 1,6 biphosphate. What is formed? Fructose 1,6 biphosphate is formed. So it is a 6-carbon sugar. It is a 6-carbon sugar. So 3-carbon, 2 molecules. One is phosphoglyceraldehyde, another one dihydroxyacetone phosphate, dihydroxyacetone 3-phosphate. This is the dihydroxyacetone 3-phosphate. Undergo condensation. Okay, they fuse and they form fructose 1,6 diphosphate. Okay, it is 6-carbon compound. So next now, fructose 1,6 diphosphate on dephosphorylation. Dephosphorylation means removal of phosphate. Removal of phosphate. Dephosphorylation. Okay, using the phosphatase enzyme. So what happens here? You will get the fructose 6-phosphate. One phosphate is removed. One phosphate is removed. One phosphate is removed. Fructose 1,6 diphosphate, what it will become? Fructose 6-phosphate. One phosphate is removed. Now again phosphate is removed. Okay. Now, what I have? Fructose 6-phosphate will undergo isomerization by isomerase enzyme, and what it will produce now? Glucose 6-phosphate. What it will produce? Glucose 6-phosphate. So these are all 6-carbon compounds. glucose 6-phosphate is formed. This glucose 6-phosphate finally undergo dephosphorylation. So, glucose 6-phosphate phosphatase enzyme. Now it will become glucose plus phosphate or inorganic phosphate, whatever you like. So this is the product of photosynthesis. This is the product of photosynthesis. So we get glucose, is it or not? When 6 molecules of carbon dioxide are mixed, resulting in the synthesis of 1 molecule of glucose. So a molecule of glucose, how many carbon compound it is? How many carbon compound there? 6 carbon compound. So these reactions occur in the cytoplasm of the mesophyll. Suppose if excess of glucose is there, that can be converted into sucrose. Sucrose is a translocating sugar. It is transported from green parts of the plant. Sucrose, sugar, table sugar, what we use daily. Okay, this is transported from green part of the plant to non-green parts of the plant. That is okay. If excess of glucose is there, it can be stored as a reserve food material. Which is the reserve food material in plant? Reserve food is starch. So glucose can be converted into sucrose, and sucrose is transported through phloem. Phloem, you know, food conducting film. Okay, from green parts to the non-green parts. The excess of glucose, when it is there, it is stored as a reserve food material in the form of starch. Okay, so these are sugar formation. That's how sugar is formed by using 2 molecules of 3-phosphoglyceraldehyde. Remember, 3-phosphoglyceraldehyde and DHAP both are triose phosphates. Right? Now, one step is remaining, that is regeneration of RUBP. And the schematic cycles on that. I will continue tomorrow.